Hello, everyone, and welcome to this episode of our Discover Molecular Interactions webinar series. My name is Vivian, and I'm the head of scientific communication and marketing at Bruker Biosensors. And this is, in fact, also the first episode of our series that we host as Bruker Biosensors instead of Dynamic Biosensors after our recent acquisition. And today we will explore a very promising therapeutic modality that is bispecific antibodies. And I am very happy to introduce our guest speaker for today, Professor Harald Kolmar. Welcome.
Yeah, thank you very much, Vivian, for inviting me.
Professor Kolmar is the department head and professor of applied biochemistry at the Technical University of Darmstadt, and his current scientific interests focus, among other things, on protein engineering and design. If you have any questions during the talk, please feel free to use the Q&A feed or also the normal chat function, or you can, of course, also raise your virtual hand after the talk and ask your question directly. And now, without any further ado, I will hand over to Professor Kolmar, and I'm looking forward to your talk.
Yeah, thank you. Hello, everyone. It's nice to have you here at this already dark November day, horrible day, but hopefully I can entertain you with great science, which is about bispecific antibodies. And as an introductory note, we are more nearly exclusively involved in cancer research and making therapeutic antibodies or candidates of therapeutic antibodies against cancer. And all of you may know that cancer is a number 2 in mortality and on worldwide on diseases. And you can also see on the right-hand side here that most problematic ones are lung, breast, and colorectal cancer, prostate cancers, and of course there's an urgent need in novel therapeutics in this area. But there are also great prospects now in the treatment of cancer. And there are many different options of treating cancer. Of course, classical ones, which are already existing since decades, like surgery or chemotherapy. But a very promising aspect is immunotherapy. And I would like to communicate to you why bispecific antibodies are very promising compared to classical monoclonal antibodies. This is now also from the prospect of pharma business a huge field. We have hundreds of antibodies in clinical trials. More than 100 are approved right now as therapeutics. And this is a listing from last year, which indicates that close to 800 antibodies are in early stage clinical trials only in cancer. And if you look at that listing, which is here, monospecific, so classical antibodies which bind a particular target are a bit more than 50%, but there are new modalities coming up, which are multi-specific antibodies, which of course starts with bispecifics, and also immunoconjugates. I don't want to report on immunoconjugates, we're also working on, but focus more on bispecifics. And if you look at this yellow section of bispecifics, you can look at it in a more detailed manner, and then you see that most of these multispecifics are so-called cell engagers. And these bispecifics and multispecifics have a different mode of action. They always have two different binding heads, here shown as a yellow one and a red one. And why this is being desired and why these are particularly interesting in immunotherapy of cancer is shown on this slide. What you would like to do in immunotherapy is to trigger immune cells, mainly T cells or natural killer cells, direct them to cancer cells in a way that they recognize the cancer cells and can induce apoptosis, so destroying those cells. And the main key player, as mentioned, are T cells and natural killer cells currently. So this can be done by bispecific antibodies, which are built up in a way that one arm always targets a tumor-associated antigen, any tumor antigen which is overexpressed on the surface of tumor cells, while the other arm targets and activates an immune cell, like here a T cell. Classical target being used is CD3 for T cell activation. So by this recruitment of the T cell to the cancer cell, an activation goes on, which results in the release of cytokines and also cell destroying proteins, which eventually result in the destruction of the cancer cells. Very similar is an equivalent strategy where you do not target T cells, but natural killer cells and K cells. And for that, you target different surface receptors, like, for example, classical one CD16, which eventually also leads to bringing these NK cells into the proximity of the cancer cell, activating them and inducing cell lysis by secretion of perforins. And also in this case, the whole process is supported by the release of cytokines, which can also directly lead to the activation of other immune cells like macrophages, neutrophils, and so on. So what you need are bispecific antibodies. And these bispecific antibodies it's quite a complicated process to make them. And we very recently, I think, made a very simple contribution to that, just to take approved monospecific antibodies and convert them into bispecifics that address immune cells. And we have worked on that with 3 different examples, and I would like, for the sake of time, I have here only focus on one. So the idea is you start with an approved antibody. This one, I will come to that in a moment, is called obinutuzumab for non-Hodgkin's lymphoma treatment, or for solid tumors, cetuximab on the market 6 decades, or this one, trastuzumab, for treatment of breast cancer. Cetuximab targeting EGFR, trastuzumab targeting HER2, and obinutuzumab targeting CD20 on lymphoma cells. And if you would like to make a bispecific out of it, at least conceptually, you have to change the setup of the therapeutic antibody, like here obinutuzumab, in a way that you can make a bispecific. So you need 2 different heavy chains at least. Yeah, your one heavy chain is the original one that binds to the tumor target from the therapeutic approved antibody, and the other one contains now at least one domain, the VH domain, that targets a new target that supports NK cell or T cell recruitment. And since this is now an antibody that has no longer full symmetry, you need to express, in this case, the 2 different heavy chain and make sure that they assemble as a heterodimer, not as homodimers, which is undesired, but this is easy to do because there are very well-known mutations you can introduce into the Fc part of the antibody that steer the formation of heterodimers. And there's also now, since a couple of years, freedom to operate to use these mutations that direct the formation of heterodimers, so-called knob into hole technology. So you have to express those 2 heavy chains, and things are going to be very easy when you are able to use one and the same light chain, which is also coming from obinutuzumab. So you would need on the right-hand side, which is new, a binding which is only relies on the heavy chain, binding here the immune cell target, and keep the light chain. It's getting more complicated if you would also have a light chain that mediates binding, but there are examples out in nature, like in camelids, that only one domain is sufficient for binding, and so you could set up an expression system where you express 2 heavy chains and 1 light chain that pairs with it in the same manner, and here it's mediates binding to the tumor target. Here there is a surrogate light chain, and binding is mainly mediated by the newly introduced, here shown in blue, heavy chain domain, which of course has to be isolated and found. And we do that by using a special technology, which I will show you in a moment. But here very briefly, the mode of action of this obinutuzumab, which is particularly important for B-cell lymphomas. These are tumors derived from B cells in the class of non-Hodgkin's lymphoma, and the first antibody that was approved among the first ones is rituximab, already approved in 1997 for treatment of B-cell lymphoma, and the follow-up antibody, which was approved 2013, is obinutuzumab, who has several additionally built-in features, making this antibody more potent. It, like rituximab, it binds CD20, but it has some engineering of the Fc part so that it, for example, better addresses macrophages. For the specialists among you, it's afucosylated and therefore more potent in binding Fc gamma receptor 3. And so we thought this might be an interesting starting point to see whether we can further improve the potency of this antibody and also direct it in a bit different direction. You may know that not only IgG antibodies, which are nearly all therapeutic antibodies are of the IgG type, not only these exist, but there are also in our body IgA antibodies. IgG antibodies can also recruit by themselves immune cells. This is mediated by the Fc part to bind to the Fc gamma receptor 3, also in immune cells, but here with a very low affinity. If you improve that affinity, you have a better potency of those antibodies. But there are also antibodies in our body of the IgA type. IgA antibodies bind a different receptor and also different cells. They mainly bind neutrophils. These are myriads of these exist in our blood, and also macrophages. So there are also ideas to make antibodies that activate neutrophils and macrophages via binding to the receptors that are normally targeted by IgA. And you can make IgA-derived antibodies, which is very complicated since they are heavily glycosylated and also have a short half-life. Of course, we would like to have an extended half-life of a couple of weeks, like in IgG. And therefore, for example, tandem IgG-IgA antibodies have been made. Bispecific antibodies have been made in a bit weird format where you have here a binding moiety for CD20, like in rituximab or obinutuzumab, and here an Fc part of a normal IgG, and here then placed a domain which binds CD89. CD89 is this Fc alpha receptor, which is the natural target of IgA. We thought, let's make it easier and have this obinutuzumab binding on the left-hand side and have this CD89 binding, like in normal IgA antibodies, at the right-hand side. And we also can play around with the affinities here to make it good. But how to get this domain? And for that, we always go the same route. We immunize chickens and later on mine the repertoire of antibodies that are being made by chickens. So in this case, we immunize chicken with the CD89, for example, but I will show you in a moment. Why chickens? There are simple reasons. Chickens are far away from humans, evolutionary speaking, 300 million years, and it's always a problem if you immunize with an antigen, an animal, which exists already in that animal very similarly, then it's hard to get an immune response because there's always a self-restriction going on our body, and also that one of animals avoids making antibodies against already existing proteins and antigens, and these are always being removed. And if the target protein doesn't exist in, for example, chickens, or is with respect to sequence far away from the one you use for immunization, this is shown for EGFR, for example, then you get a broad repertoire of immune response, and this is exactly what we see upon immunizations of chickens. And so what we did in this case is we immunized the chicken with the extracellular domain of CD89. As I said, CD89 activates macrophages and neutrophils and dendritic cells, and should then mediate and enhance the killing of tumor cells, in this case, these B-cell lymphoma cells. We immunize the chicken with this domain. We take B cells out of these chickens and isolate the cDNA encoding for these antibodies, and take out, in this case, only the DNA that is responsible for making the VH domain, because we are looking for this blue VH domain as a binder of CD89. And what we then do is we take the light chain, in this case, the VL domain from the light chain from obinutuzumab, and combine them to a single chain fragment by putting a linker here. So all of that is being placed on the surface of yeast Saccharomyces cerevisiae, and we make libraries. So every clone of a yeast cell then has one and the same light chain, which is being taken from obinutuzumab, and the VH domain, which is coming from this immunization, and all of that is presented in multiple copies on yeast cells. And so you have this library of yeast cells, and you can screen them for binders, where the binding is mainly mediated or exclusively mediated by the VH domain, which comes from this immunization. At this point, this is a hybrid of human sequence and chicken-derived sequence, but later on we can convert that into fully human antibodies with the technology I have no time to report on today. So we throw in the antigen, which is the CD89 domain, directly or indirectly fluorescently labeled, and we can do ultra-high throughput fluorescence activated cell sorting to pull out interesting binders. An example is shown here. So we normally do that over several rounds of screening, and right at the beginning we throw in high concentrations of CD89, labeled biotin labeled CD89, which can then be used for fluorescence labeling. And you see that in the first round, and we also do two different kinds of labeling. One is surface display, which is being addressed by throwing in an antibody that recognizes a tag, so-called MYC tag, at the terminus of the single chain. So you only get a signal when this single chain fragment is there. It's on the y-axis. So high expression of these single chains is shown here in this quadrant. And on the x-axis, we go for antigen binding with a different fluorescent dye. And so what you collect is those yeast cells that have high expression of the single chain and also good binding of the antigen. And there are fewer, as we can see here, in the first round with high concentration of CD89. We take out those cells, normally start with about 100 million clones in the first sorting round, and which should be sufficient to cover the full immune repertoire of a chicken heavy chain domain after immunization. Take those out, grow the cells again. So if you did it right, there should be more than before, which is indeed the case. They are then in this quadrant, and then in the next round, we go down with the concentration of the target to 250 nanomolar, 100 nanomolar, eventually at the end of everything after a couple of rounds, 50 nanomolar or even less, and always take out the best candidates and analyze them and reformat them in a normal antibody format, this newly discovered VH domain with this obinutuzumab light chain, and measure the binding in the classical way, which is biolayer interferometry. So you mobilize this antibody on a tip and dip this tip into a solution that contains the antigen, and then you get an on-rate determination and also an off-rate determination. From that you can calculate the KD and pull out the best binders. And I show you here only 3 clones we got of many from the screen, which do their job in presence of the light chain from obinutuzumab, indicating that binding is being mediated by this VH domain. And if you look here at the numbers, we have single-digit nanomolar affinities, except the first one, for these candidates. And then we can make bispecifics, express them as well, where you have now at the left-hand side the obinutuzumab binding moiety, on the right-hand side this newly discovered VH domain with the obinutuzumab light chain, and measure also here binding to CD89. And so there's not a big change in the KD, but only in the amplitude. Reason is here it's bivalent binding, here it's monovalent binding. And of course, later on, you can characterize this candidate in more detail and also check on cells whether you have a good binding, which is indeed the case. When you compare that with when you take cells which are CD89 plus, obinutuzumab does not bind there because it has no implemented CD89 binding, no FC alpha receptor binding implemented, but these candidates here, two of them are shown, have single-digit nanomolar binding. At the end of the day, of course, we do a functional test. In this case, we ask the question, can macrophages be recruited, which is indeed the case. And so when you have obinutuzumab, there's not a lot of phagocytosis that you can induce, but this is significantly enhanced with this newly generated candidate. So which indicates that there's also enhanced biological activity. And you can do that in principle with any therapeutic antibodies. This was the first story I wanted to tell you, but this is another next generation of bispecific antibodies, so-called 2-in-1 antibodies. So what are 2-in-1 antibodies? As I showed you before, a bispecific antibody has a left-hand side arm binding target A and a right-hand side arm binding target B. A 2-in-1 antibody is a symmetric antibody. It looks like a normal antibody. It has two identical heavy chains and two identical light chains, but simply speaking, the light chain binds target A, the heavy chain binds target B, both on two arms and also simultaneously. So you have a symmetric bispecific antibody that is tetravalent, twice A and twice B binding. And of course, this is a very appealing format because you only have to manufacture two chains. As always, for every classical antibody, it also looks like a classical antibody. But how to get these 2-in-1 antibodies? And we started with that a few years ago, and had the idea to combine the binding to EGFR and PD-L1 in one antibody format of this 2-in-1 antibody. And there are good reasons for that, because on several tumor cells, there's both EGFR and the target PD-L1 overexpressed. So you have if you have a combined binding, this may contribute to enhanced specificity of such type of an antibody. And other feature which is also interesting is that PD-L1 has a receptor on the surface of T cells, which is called PD-1. And this is one of the strategies of tumor cells to escape the immune response by T cells. This is overexpressing PD-L1, which then blocks PD-1. And by this PD-L1 checkpoint inhibition, T cells are non-functional and do not care about tumor cell killing. And this is why strategies exist to either block PD-L1 or PD-1 with antibodies. And if you do so, you can enhance the activity of T cells per se, which then take care of tumor cells and can kill them. Classical example is Keytruda with 25 billion US sales, annual sales now. I think this is the best-selling antibody currently on the market. So if you make a 2-in-1 antibody that also blocks this axis, then you would, in addition, have implemented this checkpoint inhibition and enhancement of activation of T cells, which would be nice. So what we did is exactly the same as mentioned before. In this case, we started out and immunized the chicken with the extracellular domain of PD-L1 and combined that on the surface of yeast with any common light chain we are always using. This goes away later on, as I will show you. This was just a surrogate. So that we once again we get a library of, in this case, Fab fragments. Before it was single chain with yeast, but doesn't matter, which can be screened for PD-L1 binders, this heavy chain library. This is what we did, and also isolated one, which looks like quite promising. With I only show you data on checkpoint inhibition. An approved antibody, which is anti-PD-L1, is durvalumab, which has an EC50 in this inhibition of 580 picomolar in our assays. Our ones, the new one, which relies for binding mainly on the heavy chain, because this is a common light chain we just threw in, is 700 picomolar, very close. So we are very happy to see that this checkpoint inhibition works. And then we started out with this PD-L1 antibody and put that on the surface, that heavy chain we isolated on the surface of yeast cells. And in this case, we also immunized the chicken with EGFR, extracellular domain. In this case, we take out the light chain diversity, and combine them with molecular biology methods just by transforming yeast and secreting it, then they automatically assemble. So now we have the heavy chain constant, which is already programmed to bind PD-L1, and add a library of light chains, of which we hoped there would be some that bind EGFR, and then you would end up with this desired format. And this is how we did it, and also found a candidate which simultaneously bound PD-L1 and EGFR in this setting. And also, as we could show by biolayer interferometry, it can bind both simultaneously. Despite the fact that these domains, of course, are close together, you can throw in PD-L1, get a layer thickness increase, and biolayer interferometry showing that PD-L1 is bound. Then you add EGFR and you get a further increase, indicating that also EGFR is bound, and you can also play the game vice versa, first starting with EGFR and then coming up with PD-L1. But as you can see, the biophysical properties are lousy, fast dissociation going on, and particularly for EGFR, disappointingly low affinity. PD-L1, okay-ish, but for EGFR, we thought disastrous. But then came the surprise when we measured these affinities on cells, particularly on cells that both express simultaneously EGFR and PD-L1. And here, our 2-in-1 antibodies displayed a very good binding. So EC50 of binding 1.4 nanomolar, which is quite good. And we also saw that this antibody had some binding left on fibroblasts and keratinocytes, which also unfortunately express EGFR and also always negative interfere with EGFR targeting for tumor therapy. But compared to cetuximab, the unapproved antibodies, this binding was reduced. So we already gained some selectivity, and obviously saw a synergistic effect of this 2-in-1 antibody when it binds cells that both express EGFR and PD-L1, which is not the case for these non-cancer cells. And we also went into that with the help of dynamic biosensors and the company 2Bind, and used this Helix Cyto biosensor, which is really a great invention. So it relies on the trapping of single cells. In our case, of course, EGFR, PD-L1 overexpressing tumor cells. And then you throw in a labeled fluorescently labeled analyte molecule, and can, in this case, of course, our 2-in-1 antibody. And then you can see the binding on the single cells which are trapped in this device. And also, since this is a microfluidic device, when you let the antibody go again, you can determine dissociation and can determine upon multiple injections the K on and K off rate. And we were very curious to see whether we get, and of course we did it in bulk before, so these measurements are just binding of antibodies to cells, and we were very curious to see how it behaves in this setting, because this is a great opportunity to determine not only the KD, but also K on, get an ideas about K on and K off. And I was very pleased to see that the KD that you can calculate from these measurements is very close to the ones we get when we do these bulk affinity measurements everybody does. And also here, of course, you gain important information, for example, how fast the molecule dissociates from the surface of tumor cells. You say this is a slow dissociation. This is exactly what you would like to see. But still, we thought, okay, it's not so nice to have a binder with 270 nanomolar affinity to EGFR. So we asked the question, can we improve the affinity? And so I would like to show you how we do affinity maturation right now and currently. What you do in this case, since this is the light chain, you have to improve which is responsible for EGFR binding. You can do some kind of randomization. So with primers and with gene assembly, we made a library of the CDR1 of this EGFR binding light chain, where each of the 9 amino acids is randomized so that all other 19 amino acids are also allowed. And the same we did in CDR3 with the primer sets, which is 12 amino acids long. So you get also here at each position 20 variation, and you can do that simultaneously so that you can obtain a library where each residue of CDR1 is simultaneously combined with any other residues at any other position in CDR3. And you get a library out of that, and you can once again do this display screening and further reduce the concentration of EGFR during the screening to go for higher affinity binders. And this is exactly how we did it, and got a bunch of candidates, and it turned out that a single amino acid exchange made it. And this was in CDR3 of the light chain, where we converted a tyrosine to an aspartate or to a glutamate. And you see that the affinity went down from about 280 nanomolar to a single digit nanomolar. And if you, these are negatively charged amino acids, when you introduce a positively charged amino acid, the molecule is dead. So that this clearly indicates that this affinity maturation can be done. And we also verified that with the switchSENSE technology from dynamic, it's no longer dynamic biosensors from Bruker, which is also allows you to determine binding. In this setting, you may know that all of you, since you are users, most of you are users of these devices, know it better than me, that you immobilize the ligand on the surface, or use a DNA aptamer strand and put a dye on, and you can do that simultaneously for both targets if you are going to be using both dyes, which is very, very effective. And so we compared the non-affinity mature to the affinity mature variant, and looked at the fluorescence change going on here. And if you look to the right-hand side for PDL1, you see with your naked eyes that there's not a big change in the signals, but for the one where you have this low affinity EGFR binder and here the high affinity EGFR binder, you see that you get enhanced signals which are related to enhanced affinity. Of course, these later on can be quantified doing this kind of measurements, and also helix cyto has been used for quantification of binding. And for example, what I find interesting, you can measure the retention time on the cell surface for the different variants and for different cell lines, expressing different levels of EGFR and PD-L1. And if you use one in the same cell line and look at the retention on the cell surface, for example, for EGFR, you see a significant improvement in this half-life by having this mutation in place, so that you get quantitative information that also allows you to see directly the improvement and to get information what's going on tumor cells and not only on immobilized surfaces, which is a great benefit, I think. So my take-home messages are that we can generate antibodies from immunized chickens. We can make bispecific and oligospecific formats. We can convert therapeutic antibodies into bispecific antibodies quite straightforward. We can also make two-in-one antibodies that are a new class of antibodies in the bispecific fields, and I think it's absolutely required that you understand in detail the biophysical and functional properties of these multispecifics, particularly in their biological environment, means on cells before you go into animals. And this is required for the development of next generation biologics. I would like to thank my co-workers who have done most of this work. This was in my group, mainly Julia Harvard. He is now at Merck in Darmstadt, and also Jan-Patrick Bogen, who is now at BioNTech in Mainz. I would also like to thank the financial support of Ferring Pharmaceuticals at the beginning of this project, and my great thanks go, of course, to Dynamic Biosensors, to Bruker, Johannes Mehring, Guido Uhlenbrock, who made these cell-based measurements, and Vera Molkin team from TrueBind, who also did in-depth analysis of binding of these molecules using the switchSENSE. And with that, I would like to end and thank you for your attention.
Thank you very much for this very nice talk. Yeah, the floor is open for questions, so please feel free to write any questions you have in the chat or the Q&A, or also raise your hand. And there's one organizational question. This webinar was recorded and you will be able to watch it afterwards. So I can already answer that question, but maybe let's start with the first question about the projects themselves. Could you maybe explain, because you showed so many technologies to measure on rates, off rates, what do you think is like the added value of doing this on cells instead of just sticking to BLI, for example, which also shows you certain differences?
Yeah, what we also see is that BLI data and on-cell binding coincidence. This is, of course, what you would like to have, yeah. But we also see, for some examples, that there are significant differences in the affinity you measure by BLI, and you eventually see on cells, because on cells it also depends on the density of the target on the cells, and when you have high occupancy, it makes a difference compared to low occupancy with normal antibodies who can have simultaneous binding with the two binding arms. You can have avidity effects, and you can only see that realistically on tumor cells. So what you get normally is just the information about the KD, but you do not get the information about how long they stay, what's the off rate, what's the on rate, and these are biophysical characters that are important, particularly when you are in a discovery process of antibodies and would like to get the best candidate. And so, and for that you need a detailed analysis. And even if you have two binders with an identical KD, it could be that one has a good on rate and a bad off rate, and the other one has it inverse, and it depends on your needs then to decide which one is the best one, and you cannot deduce it just from the KD alone. And this is why I like that much this on-cell K-on, K-off measurement, which is new and was not possible before. And I think it will be supportive for the discovery of antibodies.
Mm-hmm. Yeah, I agree, especially if you think of immune cells, right? The immune synapse is something that's very dynamic. It's not always there. It's going to be formed. Receptors are clustering. So these clustering effects are still there if you measure on the cell, right?
This is another aspect that has to be considered. It's also the design of the proximity of the two binding parts of a bispecific. We recently published that. As you said, the immunological synapse to be formed is important, which also is dictated by the distance of the two binding moieties. And you can target tumor targets which are close to the membrane or those that are distant from the membrane. And this also dictates synapse formation. I'm not aware that the technology you offer has already been used for studying, for example, the synapse formation and interaction of tumor cells with immune cells, but this could be within reach at some day. But of course, this would be a new avenue of opportunities. To my knowledge, this has not been done yet, but would be a dream. Yeah.
Yeah, that's true. We don't do cell-cell interactions yet. It's mostly cell-molecule interactions. But even with this more simplified model, we already see differences. If you can use, for example, as a workaround, look at fixed cells versus living cells to sort of see what happens if I retain the mobility versus if I don't allow the receptors to cluster. That's things that are done already that gives some information about this.
Good to hear. Great. Okay.
Okay, I do not see any further questions. If you realize afterwards that you have additional questions, feel free to reach out to either Professor Kolmar or also to us. You will find us on our website, which is still dynamicbiosensors.com. So you can find more information there. You can also find the recordings of previous episodes of webinars there. And you will also find the next episodes, which will probably start with the next year again. So thank you very much for your participation. And thank you, Professor Kolmar, again for the great talk and the discussion.
Great. Thank you for inviting me. And thanks to all the participants.
And with that, I wish you all a great rest of your day. Thank you and goodbye.
Okay, goodbye.